This is lecture 22 for Biology 177 on action potentials and neurophysiology. This handsome gentleman here is Julius Bernstein. He was actually a German physiologist during the mid to late 1800s that came up with the theories and later some of the research that showcased resting membrane potentials and action potentials in cells and how ions move. So thank you to Bernstein. So in the previous lecture, we talked about membrane potentials, including the resting membrane potential and then graded potentials. To review graded potentials a little bit, whether they're depolarizing or hyperpolarizing, they share four basic characteristics. One, the membrane potential is most affected at the site of stimulation, and the effect decreases with distance. That is, where it hits is where it's strongest, and as soon as it ripples out from that point, it will decay. Two, the effect spreads passively through local currents, like I said, through that kind of local ripple wave. Three, the graded change in membrane potential may involve either depolarization, positive change, or hyperpolarization, negative. The nature of the change is determined by the properties of the membrane channels involved. If it's a sodium channel, then you're going to be dealing with depolarization. Potassium channels cause hyperpolarization. Thus, the change in membrane potential reflects whether positive charges enter the cell or leave the cell. 4. The stronger the stimulus, the greater the change in the membrane potential and the larger the area of effect. So we've seen this diagram a few times where we have the dendrites that collect the electrical signals, the cell body that integrates those incoming signals and generates an outgoing response to the axon. The axon is going to pass electrical signals to dendrites of another cell or to an effector cell. Once the actual graded potential reaches the axon hillock, which is the base of the axon, it will automatically transition to becoming an action potential. An action potential is simply a stimulus that is strong enough to reach threshold. We can notice here in the top diagram where we see membrane potential in millivolts on the y-axis. The x-axis is always going to represent time. In the first two little bumps, we can see subthreshold stimuli. So it's just a simple stimulus that causes a slight depolarization, but it's not really strong enough to cause any sort of meaningful change. On the third wave, we notice that initial change, that initial stimuli is strong enough to reach what we know of as threshold, which is identified as -55 millivolts. Threshold is simply where the stimulus is strong enough to reach the axon hillock and open voltage-gated sodium channels. This opening of the channels causes a drastic depolarization of the entire cell, as has been illustrated in the diagram, and will then go through the action potential and carry its way back down. This threshold stimulus is key in generating an action potential. Now, if we go to the last bar on the bottom diagram, we notice suprathreshold stimulus. This is a very, very strong stimulus. The stimulus in this case was stronger than it was in just the threshold stimulus. But notice that the action potential generation is equal on both. So it might get to the actual threshold point faster, but it's not going to depolarize the cell any more than to the +30. So all you need is a stimulus that is strong enough to reach threshold to create an action potential. Going above that doesn't generate a stronger action potential. Here we can see the action potential. In your lecture notes, this is actually the diagram that you're going to draw, and I'm going to take you through each step. Step 1 is resting membrane potential. Resting membrane potential is what we've already talked about in the previous lecture. Here we are at -70 millivolts. The channels are all closed, the leak channels are open, but it's just going to be creating that kind of natural resting environment. 2, we have a depolarizing stimulus. This stimulus is a graded potential and the membrane will depolarize. Of course, you have to remember that it needs to be strong enough, and that's where we get to 3. The membrane will depolarize to threshold. Once we reach threshold, which is -55 millivolts, both sodium and potassium channels will begin to open in a sequential process. The voltage-gated sodium channels causes a rapid sodium entry depolarizing the cell, and we are now at 4. 4 is that rapid depolarization as sodium enters the cell, and it gets all the way up, and it can get all the way up towards positive 60. Next, we go to 5, which is sodium channels close and slower potassium channels open. Here, the sodium channels are inactive and it stops moving across the membrane. We have reached peak action potential. It's done its job. From there, we move to 6. The potassium is now going to open and move from the cell and into the extracellular fluid. This is going to cause a drop in the actual membrane potential of the cell, making it more negative. This process will continue as it goes down and reaches 7. Here, the potassium channels will remain open and additional potassium will leave the cell, hyperpolarizing it. So it's actually going all the way down below the resting membrane potential. In this case, as low as negative 90 millivolts. From here, the potassium channels will shut and the natural leak channels will balance the situation out as potassium is going to less potassium will leak out of the cell, but sodium is going to leak into the cell, and it naturally will restore itself to resting membrane potential at negative 70 millivolts. So that's going to be the overall process of the action potential and all the steps that it goes through. So in the previous diagram, what we actually looked at was simply the voltage level change and diagram of an action potential. Here is the actual movement of channels during those different events. So we'll start at resting potential. At the resting potential, this is our natural environment. Sodium is high concentration on the outside of the cell. Potassium is on a high concentration on the inside of the cell. The only channels that are open are going to be the leak channels. Right now, those gated channels are going to be closed. We then get a stimulus that is strong enough to depolarize to threshold. From here, we can see a rapid movement of sodium as it starts to move into the cell, and these sodium channels the sodium channels will open, and then it goes from 1, depolarization to threshold, to 2, activation of those sodium channels, where it rapidly is going to move into the cell. This is going to make it a lot more positive. As it gets more positive, it's going to keep moving into the cell until it reaches up, typically about positive 30, closer to that equilibrium potential for sodium. And from there, it will shut the sodium channels and open the potassium channels. We notice potassium in step 3 is leaving the cell. By potassium leaving the cell, it's going to make the cell more negative. And from there, it's going to keep leaving the cell until it reaches down to negative 90 millivolts, where it hyperpolarizes. Here, it is going to close the channels, thereby allowing the cell to return to a resting membrane potential through the actions of the leak channels. So these are the actual physical events that go along with that electrical diagram from the previous slide. Now this might seem random, but we're going to talk a little bit about local anesthetics and where and how they work. So what do they actually do? Local anesthetics, things like Procaine or Tetracaine, lidocaine, stuff like that, they act directly on the sodium gates and make them less likely to open. They essentially are going to plug it up. So if we notice here in this diagram, we have a natural open sodium channel where sodium can flow through. Then we have an example of a closed sodium channel. We can also, in the cases of anesthetics, plug that sodium channel. So if we plug the sodium channel with these different anesthetics, it's going to block the movement of sodium in, so therefore we can't transmit any sort of action potential. That means you're not going to feel anything from it. Now, many of these anesthetics actually degrade over time. So say, for instance, you go to a dentist office and you're getting your wisdom teeth removed. You'd actually take the injections, you wouldn't feel anything in your jaw, and then several hours later, you're going to start to feel it wear off. This is because your body is breaking down that anesthetic and the sodium channels are unplugged and working again. Now, when talking about action potential generation, there is a refractory period of that action potential. There's an absolute and a relative. The absolute refractory period simply means this is a time period in which no other action potential can occur, no matter how much stimulation happens. A relative refractory period is a time during which the membrane can respond. So you can generate another action potential. However, it must be stronger than the initial action potential. It's got to be stronger than the first one. So let's take a look at where that occurs. So let's talk a little bit more structurally. Once the action potential reaches the end of the nerve axon, what stops the action potential from reversing direction and not making it a one-way street? How we do that is largely dependent upon the double gating of sodium channels. When the cell depolarizes initially, two things happen to the sodium channel. One, the activation gate opens as soon as the membrane reaches threshold. This allows sodium to enter. The second thing that happens is the inactivation gate begins to close. So it's got this double gating mechanism. However, inactivation gates close very slowly. Eventually, more and more inactivation gates close while more and more potassium channels are open, so the cell repolarizes. So we can see in this diagram that sodium is entering the cell once the cell reaches threshold. This is going to be that initial opening of the activation gate. Once we actually have the inactivation gate open, that's going to be through the relative refractory period timeline. So the absolute refractory period is where the inactivation gate is closed. That's that absolute refractory period. The inactivation gate opens when we actually go through the relative refractory period. And you can see that's when potassium is leaving the cell. And during this time period, the membrane will generate an action potential if it is stimulated by a relatively large graded potential. So it's got to be something stronger than the initial stimulus. So now that we've discussed different aspects of the action potential, I want to talk a little bit more about how it moves. There are two major features that allow action potentials to actually move faster. Okay? They're going to be myelin sheaths, which we'll talk about in a bit, and the simple size of the axon. One of the first things is that large axons offer less resistance. Here we can actually see a squid's giant axon of 0.8 millimeters. That is huge. We have a nerve cell in it. There's a nerve cell in a squid that is almost a millimeter in length. So where you can actually physically see it, and it has actually been tested upon. And it was many of these tests that let us understand a little bit more about neurophysiology. Now you have to ask yourself, from this conduction of action potential, does it degrade over time? Because I said large axons offer less resistance. So does that mean a smaller axon degrades it? No, not at all. In fact, the action potential at the end of the axon is the exact same action potential, strength, everything, as the start of the axon. This is because that the axon membrane contains all of the same voltage-gated channels that contribute to the action potential. As one area of the axonal membrane depolarizes above threshold and creates an action potential, the electrical wave is going to spread out, and it stimulates voltage-gated sodium channels on the next adjacent part of the neuron. So it's going to work just like firing off little dominoes in a sequence, thus regenerating the action potential. It's going to keep regenerating back to its initial stimulus. In this manner, the action potential continues moving as a wave down the axon towards the end, constantly regenerating. Now remember, I said the size and diameter of an axon is going to contribute to the speed within which it actually moves. The other factor that affects the overall speed of the action potential is whether or not the axon is myelinated. This is a little reminder of the two glial cells that are actually responsible for myelin. The first is going to be in the central nervous system that we can see here on the left. It is the oligodendrocyte. It's going to offer structural support as well as generating myelin in the central nervous system. On the right, we have Schwann cells. Schwann cells are throughout the peripheral nervous system and create large kind of jelly-wrapped portions of the axon. Why is myelin important, and how does it help things move? Well, myelin, quite simply, allows an action potential to jump from section to section. If you can imagine, an entire axon is covered in voltage-gated sodium and potassium channels. Well, every time a voltage-gated sodium channel activates, it's going to activate the next one in sequence. Like I said, dominoes from before. Myelin will actually wrap around portions of the axon and allow that action potential to essentially skip an entire section of voltage-gated sodium channels. So it's going to bounce from area to area to the areas that are not myelinated, which are called nodes of Ranvier. As it bounces through, as it keeps kind of hopping from each one, just kind of passing through, it is going to conduct this action potential faster. This process of moving from node to node is called saltatory conduction. and it greatly speeds up the velocity of action potentials. So we've talked about how action potentials are created, maintained, and conducted. But what happens when we actually have that action potential, that wave of depolarization, when it reaches the end of the axon terminal or nerve terminus? There's very few neurons that actually have a physical connection. They typically have space in between them, gaps with other cells. So how does an electrical signal move to the target cell? It does this by converting that electrical signal into a chemical signal. So it does this at synapses, where one neuron meets another neuron or an effector cell. And this allows for the continuation or nerve impulse beyond the end of one axon. Now, there are two types of synapses, a chemical and an electrical synapse. Here we can see the events occurring at a cholinergic synapse. This is going to look very similar to the events at the neuromuscular junction, and this is the example of a chemical synapse. Here, an arriving action potential is going to depolarize the axon terminal. From there, voltage-gated calcium channels open up, calcium rushes into the cell, and the calcium will drive the vesicles of acetylcholine to leave the cell through exocytosis and then cross that synaptic cleft via simple diffusion. From there, it will bind to the acetylcholine, that ligand-gated receptor on the postsynaptic membrane, and open the channel, thus allowing sodium to move in. From there, step 4, the acetylcholine is going to be broken down into acetate and choline. Now, the body is going to remove the acetate and reabsorb the choline. The choline is going to be repurposed to more acetylcholine through the actions of the mitochondria and acetyl-CoA. Thus, it's going to create a renewable cycle of chemical neurotransmitter. The binding of the neurotransmitter to the receptor of that postsynaptic cell can cause a variety of intracellular responses. It can open or close the channels, it can turn on signaling cascades, etc. In many cases, the end result of the actions, especially in the central nervous system, is to affect the membrane potential of the postsynaptic cell. And it can do this in one of two categories. Either excitatory postsynaptic potential, where it's going to depolarize and shift the actual membrane potential of the postsynaptic membrane to be more positive, or it can inhibit. Inhibitory postsynaptic potentials are graded hyperpolarization. So this is going to be inhibitory and open chemically gated potassium channels, thus shifting the membrane potentially further away from threshold. So in order to actually excite the membrane, you're going to need a larger than normal action, or stimulus rather, to generate an action potential. So here we can see the actual diagram of how those graded potentials from the presynaptic neuron are going to affect the postsynaptic. We can see that there's a depolarizing stimulus, and this is going to facilitate a greater overall membrane potential, thus allowing it to depolarize more easily. However, on time 2, where we inhibit, we hyperpolarize, thus activating the chemically gated potassium channels, making the cell more negative. So this is going to inhibit the cell's ability to be able to actually generate an action potential. Now, if they're both applied, we get what is called summation. And summation causes no movement, to where we essentially they cancel each other out and we are at resting membrane potential. Now, we've been showing one neuron interact with another neuron. This is actually more along the lines of what it would look like. We have a neuron with hundreds, if not thousands, of connections from other actual neural cells, to where we have all of these cell processes extending in. We have the dendrites coming out. We have all of the axon terminals coming in. So it creates a wiring system that allows for multiple cells to interact with multiple cells. And this means that we can actually have different types of synaptic activity and postsynaptic responses through a process called integration. So from that previous picture, that previous diagram, we can see that there's often many types of presynaptic neurons covering and converging on a single postsynaptic cell. The response that occurs in the postsynaptic cell is then the summation of the inputs of excitatory postsynaptic potentials and inhibitory postsynaptic potentials from that presynaptic cell. Now, if enough excitatory potentials are generated, you can reach threshold and create an action potential. But each inhibitory potential makes it harder. So that means, and because when we have all of this combination from these different neurons, we call this summation, as we said in the previous slides. So there's two types of summation. The first is temporal summation. Here, graded potentials happen rapidly from the actual same axon terminal. And if they happen rapidly, they can combine to make a larger change in the membrane potential. We can see that here. We have a first stimulus coming from an axon terminal, and then a second stimulus that makes it a little bit stronger, strong enough to actually reach the axon hillock and generate an action potential. This is temporal summation. Spatial summation is going to be excitatory or inhibitory action potentials that can combine to form larger depolarization happening from separate axon terminals. So we have two separate stimuli. So these axon terminals are going to each generate their own excitatory or inhibitory, and they summate together. And in this case, it was excitatory enough to where it was able to generate an action potential. And that finishes our covering of the action potential lecture. In the next lecture, we're actually going to get into the anatomy of the brain.